Welcome back to the LundX channel that make your research simple. And this is the last part of single cell RNA-seq analysis, in which we will learn about how to find markers in different cell cluster and cell annotation. You know, this is the most important part of single cell RNA-seq analysis. Let's learn about it. In last part, we learned about UMAP and TSNE. So before we are going to run a find marker function from curate, we need to join layers. What happens? It will join layers for all markers from all cluster. So we need to run this join layer. And after that, we need to find marker function from curate. So just like if we want to find a markers from cluster 3, we need to give the name of that curate. So just like if we find cluster number 2, and here we need to give a cluster name number 2. So find all markers from cluster 2. Okay, let's run it. It has been started, so it may take some time. We need to wait for it, as calculation has been started. So once it finish, just like here, it has been shown as 0%. When it become 100%, it will be finished. So we need to wait. Okay, let's check. It has been completed. Here 100% is shown. So now what we did, we find cluster markers in 2, cluster 2. We find markers from cluster 2. Here look, in environment it has been uploaded. So if we want to check, or just if we want to check, suppose top 5 or top 10, we can use the head function from curate, because we gave a name of curate just like a cluster 2 marker. So we need to use a head function and give a name the file, and then we give a number of features or genes or markers. So if we want to see top 5, we can run this one here. Okay, now second option, if we want to save these all markers, then we need to save by using a function write scb, and we can give a name is cluster 2, because we find a cluster 2 markers, and here we also give 2, and we give a file format as csv. Let's run it. Okay, we want to check whether this file has been saved or not. Okay, here is the cluster 2 markers, and file save in Excel sheet. So okay, if we want to compare a marker of any specific cluster, for example cluster number 5, in comparison to those clusters which we have doubt that marker of these clusters are somewhat related, especially when we look at UMAP, there are some clusters which are connected with each other. These indicates these are related cells, so marker of those clusters may have some similar. So in that case, if we want to compare marker of a specific cluster in comparison to other specific cluster, for example, if we want to compare cluster marker of cluster 5 in comparison to 0 and 1, so we use the find marker function from curate, and to find a marker of cluster number 5, when we run this function, it gives those markers highly specifically expressed in cluster 5 in comparison to cluster 0 and cluster 1. So this function can be used when we have a doubt between some cluster and the marker of those clusters may have some related or similar. In that case, we can use this function and to identify those markers specifically regulated in the specific marker in comparison to other markers. So for that purpose, we need to run this function from scRNA, just like find markers and give a cluster number 5, and in comparisons to, we also mention just like 0 cluster and cluster 1, and by using this function we can easily get the markers differential express. Let's run it. So it has been finished now. It just like it is going on. Here is saying that calculation 0%. So once it is completed, and then we can check. So let's we need to wait. Okay, let's check. It has been completed, just like it has been shown as 100%. So it has been finished. Now either we want to check directly or we want to save. It's depend on you. For example, if I want to check, I use just like here is curate cluster 5 markers, so I use a head function from curate and give a curate name just like a cluster 5 markers, and I want to see top 10 or top 20, for example top 10 markers from, okay, I want to run. Here is the top 10 markers which from cluster 5 curate. So I want to save using this just like write CSV function from curate, and cluster 5 markers is it has been loaded in curate, and I give name is cluster 5 markers in comparisons to 0 and 1. So I want to save, so needs to run. Now let's check cluster 5 markers comparisons to 0 and 1. Okay, so let's check here cluster 5 markers comparisons to 0 and 1. This is the file in which the markers only presents belongs to cluster 5 in comparisons to 0. These are the markers only present in cluster number 5 in comparisons to cluster 0 and 1. So after that, now we want to find markers in all clusters. So you can use this function find markers. From all find all markers. It means it will find all markers from all clusters. So, but in in my case, is I already repeatedly mentioned that my laptop is not very good, so I don't want to use function find all markers. Instead of that, first of all, I want to select a top 100 or top 50 or top 500 variable features, and then I find all marker functions from those 100 or 50 marks. But in your case, you can directly use find all markers from the curate. So in my case, I want to first select 100 top variable features, top 100, and then I want to select find all markers from those 100. So if you have large data set, you can use this function, and even in that case when your laptop is not working well. So I select just like 100, even though I want to select 50 because it takes a less time. So just for demonstration, I think it is enough. But I also give a script for you, is you can use find all markers from all clusters. But for demonstration, I just select 1 to 50 variable variable features, then I use the find all marker function from these 50 variable features. So let's run it first. Okay, now we need to use find all markers. When I run now this one, it will find all markers from these 50 variable features. It takes less time and it is enough for demonstration. Okay, now it has been started. If we look at here, calculating 0, cluster 0, so it has been started from 0 cluster and up to 18 cluster. Even though I use 50 markers, but my laptop may take some time, so we need to wait for that. It has been completed, just like cluster calculating cluster number 19. It shows 100%, and from 0 to 19 it becomes 20. So now if we want to save these all markers, similarly we can use a combined marker. Just function writes a CSV function from curate and give a combined markers, just like we find a combined markers from all clusters, and we give a name as all markers and format CSV. Let's run it if we want to save. Okay, now we want to check. Here is all markers. So after that, now what next step is that we want to differentiate. We want to divide these markers by a cluster. Use a group by function from curate, and then we give a condition that all those markers should have log2 FC value more than 1%. So more than 1. So for that purpose we need to run this function from curate. Okay, after that, now if we want to save the markers by giving a name, just like log2 FC value more than 1%, we want to save. Okay, now if we want to check, here is the log2 FC value more than 1%. These are those markers which are divided by groups and have log2 FC value more than 1%. Look at this, and this is cluster from 0. If we go down 2, 3, so on. Okay, now we want to run this function to find the top 10 markers for each color, so we need to run this. This is because combined markers is already in curate, and we can give a divide by colors, and the condition give a log12 value should be 1, and number of markers should be 10, and name of this top 10. So we need to run just like it is here. You can see top 10 markers, and now we want to save by giving name as top 10 markers of all colors. We can save it, and we check whether it has been saved or not. Here is the top 10 markers of all colors, and after that, now we want to check suppose top 10, top 10. Okay, so now it has been started. Once I finish, till we need to wait until it is finished. Okay, let's check. It has been completed, so a heat map has been shown, but because there are so many clusters in the top 10 markers, so there are number of markers are more, so they are overlapping. For that, in that case, we can decrease the number of markers for easily or clearance. If we want to save this, we can use just like this one and save as PDF, and we can use a portrait. Suppose we use rplot top 10 genes, we can save it. Okay, now it is converting into PDF and portrait, so this will be saved. We need to wait. Okay, let's check. We need to check our software here. This is the rplot top 10, and here because it is shown that it is overlapping. If we decrease the number of genes, it will be clear. So because I don't want to repeat, you can decrease just like if we write here 5, maybe it become clear. Let's learn about some tests which is specifically used for differentiation of marker of colors, one in comparison to other colors. It's up to us which we specify the colors name. And just like RAC test, abbreviated is receiver operating characteristic, a classification test. It gives us those markers which specifically differentially express in a specific color which we mentioned in comparison to other. It is based for differentiation of marker of colors, any colors in comparison to other colors. So for that, we must give a color surrogate name just like color 0 markers, and use a find marker function from surrogate and give a color 0 and threshold 0.25. Test use as a RAC, and only positive indicates only upregulated markers and the color 0. For that purpose, if we run this function, it gives markers of color 0 which highly upregulated in the 0 colors in comparison to all other colors. So let's run it. So it has been just like run. Now if we want to save these markers of the color 0 base, just like we give a name and save it. Let's check. Here it's not still finished, so we need to wait. Once it's finished, we again check it. So we need to wait once it's finished. Okay, just like it is still going on. As I already repeatedly mentioned that my laptop is slow, so it started a little bit late. So once I finish, we need to check. We need to wait till finish. Okay, let's check now. If we look here is the marker of color 0 based on RAC test. So that's how. After that, if we want to observe specific markers across different colors, these markers for example, in my case, I obtained these markers from different literatures, from different sources. Just like my sample was endometrial tissue, so I find markers of the cells of endometrial tissue. So I want to run these markers and observe these markers express in which colors. So for that purpose, I give all markers, mention all markers, and also mention the color just like blue and red, and mention is dot plot 1 and dot plot function from surrogate. So just like a color is I already mentioned. So let's run it. It has been started. Once finished, which we need to check. Okay, we need to wait. Okay, let's again check it has been finished. So now we want to save by using a gg save function from curate and give as a dot plot for a specific markers. Okay, let's we add some other words just like dot plot for a specific markers all. So let's run it. Okay, now we need to check. Once it finished, we need to check. So let's check it. Here is still not shown, so once I finish we need to again check because we need to wait once I finish. Okay, let's check again. Here is the dot plot which is being shown for all markers. So open it. If we look at here, there are markers which specifically express in a specific colors in comparison to other colors. So this is how first we collect markers from different sources. Then we check those markers express in which colors. This dot indicates average expression, just like and this color indicates average. There is also automatic procedure which can be used, but and this is manual, which is easy, and you can differentiate. Then first you use automatic, then you use a manual. Okay, let's learn about other functions just like cluster annotation. First of all, we want to visualize markers. So I want to visualize these two markers using a volcano plot, just like TACSTD2 and IPCAM. So use a volcano plot to observe these markers among different colors. Because I know these are specific markers of some cell, just like TACSTD to only luminal epithelium and IPCAM is epithelial cells. So it has been started. Now we wait till volcano plot is being shown here. So I need to wait. Okay, it has been shown just like a volcano plot. TACSTD2 express in cluster 8 and IPCAM also express in cluster 8. Basically, IPCAM represent TACSTD represents the luminal epithelial cells and IPCAM represents the epithelial cells because endometrial tissue have two types of epithelial cells, just like glandular epithelial cells and luminal epithelial cells. So IPCAM represents the epithelial cells and TACSTD2 represent the luminal epithelial cells. Both indicates cluster 8. These two markers give a confirmation that this 8 mark cluster number 8 is the epithelial, and among between two types it's the luminal epithelium. So now let's run another function feature plot. We want to observe these markers. So we need to wait. Okay, it has been shown that just like these both markers express on only in one color. So both markers have one location of expression using feature plot of using a UMAP. So just like here, so now we need to give a rename that only that colors because I don't want to annotate all of the colors. I just only use this colors. I need to give new colors ID and is character function from curate and run it. So after that, to the curate here because we already create IDs. Now we need to give a name. This just mistakenly written, so we need to just run this one. Okay, and now we want to make a dim plot to see Okay, this is the colors which has been renamed as we give as luminal epithelial cells. Similarly, this way we can annotate all of these colors using these functions. So this is the last part of single cell RNA sequence, and for upcoming videos for more analytical tools, applications, and for more techniques, you need to subscribe my channel. If you find that these videos are helpful for you, and again I want to say, if you have any question related to single cell RNA-seq analysis, you may contact me directly. You may mention in comments. I will reply you. Thank you very much, and I would like to say thank you throughout this journey.